Okay, hello learners. So we'll be waiting for like 5 minutes past three to actually officially start the meeting. Till then, maybe people might be joining. And as I can see, the strength has gone significantly down from the last time that we met. So, yeah, probably you people are anyway very busy with your lectures as such and your assignments. Nonetheless, we'll be starting at around 3:05 if people are still joining. Otherwise, maybe we'll start in a couple of minutes. For people who are just joining, we are just waiting till 3:05 for all of your colleagues to join, and then we'll be beginning with today's session. Okay, so maybe we can start with discussing how we'll proceed with today's session. So, my pattern, I've kept it more or less similar or consistent with how I actually dealt with problems in the first half in the previous meeting also. So, in the first half, I have a week two previous year assignment to deal with, and there are certain queries or doubts that I hope to basically clear through those questions, some of which might pertain to cloning vectors or more such questions, right? And plus, you will have more problems that you might have solved, which might help you in the final exam for this course. In the second half, I have prepared, I have gone through again the discussion forum and certain problems that people were facing. And I feel that I don't think many people actually read the discussion forum, but you can always read what problems your colleagues are coming up with, because it might address a question you might not even have had. Right, so you get to come up with, you get to solve new problems. You might not have originally had thought of that problem, and it might lead to a concept clearing for that particular problem, right? So I have addressed a few of those in the second. I aim to address a few of those in the second part of today's meeting. So that is how we'll proceed. If we still have time left, I would love to do week three's previous year's question. So I have prepared another presentation for that. But only if we do have time will we address that. And I'm still coughing. I'm really sorry for all the disturbance that might be causing. So it is 3:05. I think we can begin with our presentations for the day. And if people will join, uh, I'll keep admitting them as they join, right? So let me share the screen. I would need some input when I'm sharing my screen, whether or not that screen is visible. So please do give me that input when asked for. And if you people have any doubt, then obviously you can ask those as well. Um, okay, so we'll start with week two's assignment, which was basically the previous year's week two assignment. Right. So now I know that you people have also submitted your week two assignment and you have gotten solutions for that also. I also saw that there were some problems with people with solutions for those particular problems. I ran out of time while preparing the presentation. So I could not keep them in my presentation, but if we have time, we'll surely discuss them. So can somebody let me know whether the screen is visible and when I'm switching between screens, whether it is visible? I will have to just keep saying if there is no input. So I will not really know whether it is visible or not. Or maybe I can join from another device. Hold on, people are trying to join. You will have to give me some input whether or not the screen is visible, which is how I'll know whether or not to proceed further. Um, and to keep a check on that, I'll try to join on another device. Okay, I will assume that the screen share is working fine. And I'm going to basically share my screen again and I'm going to begin with the assignment, right? Okay. So beginning with the previous year's assignment for week two, let us discuss a few questions over here. The first one being, I'm so sorry, I gave out the answer. The first one being, the schematic diagram shown here represents a segment of the genomic DNA harboring a gene and the corresponding mRNA. hold a moment, and the corresponding mRNA transcribed from this region. Various elements of the gene, such as exon, intron, transcribed region, and the start and stop codons are indicated, right? Three functional segments of the mRNA are marked as A, B, and C. Which one of the following option is correct with regard to the three functional elements identified in the mRNA? So gDNA basically stands for genomic DNA. So this is a genomic DNA sequence, and it has basically certain markings, and then there is an mRNA that is presented in the 5' to 3' orientation, and then that is represented. Now, from the options below, we have to find which of the option actually matches whatever description and illustration is given over here, right? So the first option being A, B, C identify the exonic regions of the gene. So is it correct? Are A, B, and C the exonic regions of the gene? We'll find out. The second one is A, B, and C identify the 5' UTR untranslated region coding segment 3' UTR of mRNA respectively. Just a moment, let me just admit people. Okay. Uh, so they say that A basically represents 5' UTR and coding region. Sorry, A is basically the 5' UTR, B is the coding region, and C is the 3' UTR. The third option says A, B, and C identify the 5' cap region, exon, and poly A tail of the mRNA respectively. And the final option is all three segments A, B, and C represent the exonic regions of the gene. So the correct answer, option over here is the C1. Obviously, there are parts of exons that are present in A, B, and C, but as you can visualize over here, A does not really, let me use a marker for this. A just is still this region, right? It leaves behind this particular region which also contains the coding segment, so it cannot be an exonic region, right? Or it could not just be a 5' UTR region. It is basically the 5' cap region that is present ahead of the exon. Then B is just the exonic region or the coding segment. So this is correct in all three options actually, A, B, and C. And in C, it is a region which contains the poly A tail. Why? It
Hello,
does
ma'am.
not contain the coding region. Hello? Yeah.
Screen is not
Yeah, yeah, yeah.
visible.
Okay. That is what I wanted to ask earlier. Just a moment. Maybe I'll share my screen again and you people can tell whether it is visible this time or not. Is it visible now?
Yes, ma'am.
Okay. So I gave away the answer for the first question. I don't know whether you were able to follow the question as such or not. So in this question, there was basically a small description that was given. This was the description that was given. And then this was an illustration. And according to this illustration, we had to find out what did the regions A, B, and C basically signify. Right? So I was just discussing that this part A is basically the cap 5' cap region, right? Why do we say it is the 5' cap region? Because it is basically, it does not include the exonic region that is present over here, right? There is a gap. It is just the region from this part to this part. Also, it lies before this AUG or the start codon, right? So this will be the 5' cap region. Before this part is the 5' UTR. right? Then this B, it basically signifies an entire axon, which is this particular axon, right? So this option was given correctly in all ABC, but then again C is the poly tail region, right? Why is it the poly tail region? Because 3 dash UTR region lies beneath it, and it is basically a region after the stop codon, right? So over here in this genomic DNA sequence, you see that ATG, which in mRNA would be AUG or the start codon, is mentioned, and TAG, which in mRNA would basically be UAG or the stop code, one of the stop codons is mentioned, right? So only the region between the start and the stop codon will be the coding region. Anything before or after it will not be the coding region. But over here we can say that since this region is retained in mRNA and it comes after the untranslated region, this will basically signify the 5 dash cap region and the 3 dash poly A tail that is present in the processed mature mRNA. So we'll be moving to question number 2, which basically says that Sonali and her husband Ramesh are phenotypically normal, right? They are phenotypically normal, while Sonali's father was affected with an X-linked syndactyly condition called webtose. So syndactyly basically means that there is no gap between digits, right? So they are fused. That means syndactyly called webtose, right? Then Sonali's first child Rohan is also affected by the X-linked phenotype. Sonali and Ramesh are expecting their second child. What is the probability that their second child will have the syndactyly condition? Now, uh, over here we have to basically keep this in mind that since Sonali and her husband Ramesh are not affected, and this is an X-linked condition. So what kind of a disorder will it be? What kind of an inheritance will it be? Will it be an X-linked dominant inheritance, or will it be an X-linked recessive inheritance? So what is the thought process? I'll answer if nobody answers. Okay, let me only answer. So this is basically going to be an X-linked recessive inheritance. Why is it going to be recessive? Because the parents are phenotypically normal, right? So if none of the parents are affected, but the child is affected, then it is going to be a recessive condition. And over here, we say that Sonali's father was basically affected, right? So we can say that if this is Sonali and she's married to Ramesh. Okay, I don't know why these extra lines are coming up. I have to admit somebody in the lobby. So just tell me if after that the screen sharing is still working. Screen, I hope the screen is visible and whatever I'm typing is visible. If not, please inform me. So this is Sonali, the circle, and the square basically represents her husband Ramesh. And we know that Sonali's father, oh sorry, I apologize. Uh, Let me just draw I will be unable to draw something over here. I think. See, okay. Say this is Sonali and this is her husband, right? They are linked together with a married line because they are basically married. And Sonali basically had parents, and her father was affected by the disease, whatever disease they are talking about, which is basically X-linked syndactyly condition. Now, Sonali and Ramesh themselves are not affected, right? These people are completely normal, and they have had a son earlier who was also affected by the disease, and they are planning to have another child. The sex is not known of this unborn child, and they want to know whether this particular child will be affected or not. This particular child, right? So, since the parents don't have the condition, but the previous generation showed that condition, and the next generation is showing that condition, this means that this is basically an X-linked recessive disorder, right? Now, if this is a recessive disorder and Sonali's father actually had this disorder, it is probable that Sonali is a carrier, right? So we have put in this dot in the circle representing Sonali because she is going to be a carrier. Why? Because she has two X chromosome. One came from her mother and one came from her father. Right? The one that came from her father was expressing the syndactyly gene. So that is why she is the carrier, but not affected because this is a recessive condition and it needs two alleles or two copies to be affected, right? Thereafter, they have had a male child, right? This male child is also affected, which means, and for a male to show an X-linked recessive disorder, it just needs to have one copy. So there is this hemizygosity that is there in males, right? There is this condition hemizygous. We study homozygous and heterozygous, but in males, in case of X-linked inheritance, there is this condition hemizygous, which means that even if it has one copy of the affected chromosome, it will basically show the disease because the other copy is anyway going to be the Y chromosome, right? Now they basically want to know, Sonali and Ramesh, whether this child, what is the probability that this child will have the disease, right? So now the solution given to this in the previous year's question, a solution was actually incorrect. It basically mentioned this as a correct option, but I will reason it out why. According to me, this is the correct option, that is B, because if the sex of this particular child is basically male, right? If this unknown, okay, I'm sorry, if this child turns out to be a male, right? Then what will happen? Oh God. If this child turns out to be a male, what will happen is that there will be a 50% chance that it gets this X from the mother, and there will be another 50% chance that it gets this other X from the mother, right? So it is 50% possible that this child, if it is a male, he will be affected, right? But if this child is a female, right, then anyway one of the X will be coming from the father, which we always consider to be a normal individual. Wherever you are whenever you are married in, this individual is always considered to be a normal individual, right? So if this individual is we are considering as normal, this girl child could have 50% probability to get one normal X or one syndactyly linked X, right? Even if it gets the syndactyly linked X and the other X hair from the father is normal, then there is 0% chance that the female child will be having this condition syndactyly. She will just be the carrier for that condition, right? So this is the correct answer for this question. It could be argued that the father might also have carried an X, and that is how the female child would be affected. But if the father would have a defective X or an X with a syndactyly syndrome linked to it, then the father would also have shown the disease condition. So I hope I reasoned it out well. And according to me, this is the correct answer. Moving ahead, we'll move to question number 3, which basically says that given below are some of the commonly used pedigree symbols. choose the correct the option which gives the correct identification for each symbol, right? Also, when you are faced with some such questions in the exam, also in the assignment, also, and in any other maybe competitive exam that you might be sitting in after a year, a couple of years, like net, gate, etc. So always keep in mind that you actually read that this is one and this is two, not this is not one and two like this, right? Because you might know the answer and you might mark it incorrectly. It has happened several number of times with several number of people. So be aware of such things also. Anyway, the first one is an unaffected male, right? So first we have to choose where B, C, and D have this unaffected male option in the first case, right? The second one is a proband male, right? Because it is a male, it is a square box, and because it has an arrow next to it, it is basically a proband male. So, option number B and D still say that it is a proband male, right? Then the third one is basically a deceased female, right? So this female is no longer alive. So this is a deceased female, and from this only we have got our answer that it is D. But still, we'll refer the final symbol, which is basically that the gender of the individual is unknown, right? And just a simple example of what I was trying to say, if we would have read it like this, this was the unaffected male, this one is a deceased female, this is a proband, and this is a gender unknown. There is another option that is C, which is basically mentioned in such a way, right? So even after knowing the answer, people sometimes mark C as the answer because they have read the serial incorrectly. So moving on to question number 4, and just a moment because I think I need to admit somebody to the meeting. Yeah. Okay, no longer there. Okay. So the question number 4 basically says the pedigree shown below represents the dash pattern of inheritance and the red arrow marks the dash. So the easier thing is that first we answer the second dash, which is what is the red arrow marking? It is basically a carrier female, like we just went through in case of the second question, where Sonali, I think, was the carrier female. So any one of B, C, or D could be correct, right? Because it says the second dash as carrier female. No, just B and C could be correct because those are the only two correct ones for this particular blank. So the first blank is what is the pattern of inheritance? We have two options. Either it could be autosomal recessive or X-linked recessive. Now, over here, just by seeing the pedigree, you can first of all predict that this could be an X-linked inheritance because the only individuals that are actually affected are males and the only carriers are females, right? If it would have been an autosomal recessive condition, equal proportion of male and females might have been affected, and there would have been some males also who would have this carrier property, right? But since only females are carriers over here, right? And males cannot be carriers for X-linked recessive diseases, and males can only be affected, right? So over here, we conclude that X-linked recessive is the kind of inheritance that is present, and this particular arrow represents a carrier female. Usually, whenever we are solving a pedigree, we always try to write the possible or probable genotype and then proceed further. But sometimes these pedigrees are very apparent. if you can make a 100% bold choice or correct choice based on just looking at the pedigree and rule out all other options, then you are good to go. Otherwise, I would suggest writing down the genotypes. Okay, the fifth one. The fifth one says, given below is a pedigree that shows incomplete penetrance with an early onset phenotype. This pedigree shows dash mode of inheritance with dash penetrance of the disease phenotype, right? So one hint in this question is the word penetrance, right? So penetrance, when do we talk about penetrance? Is it a concept dealt with in case of a recessive disorder or a dominant disorder, right? So over here, if your concepts are very clear, you will know that penetrance is something that is dealt with when it is a dominant disorder, right? Especially in case of autosomal dominant disorders, we talk about penetrance. So over here, this kind of disorder will be autosomal dominant and not recessive, but we will also look at the pedigree to prove that how is it possible, right? So over here, if it would have been if it is a dominant or recessive disorder, it might confuse, what might confuse you over here is this particular part, right? That is the second row and fifth and sixth individuals, right? In the second generation. So these individuals are basically unaffected, but they have affected children, which is something that we read in case of recessive disorders, right? But you have to be smart. So just like I told you how to deal with questions, because it talks about incomplete penetrance, it has got to be a dominant disorder. So we'll consider this as an autosomal dominant, because in autosomal dominant also there are such conditions wherein the parents might not show the disease, but the children are affected, right? But in such cases, the parent would have the genotype for the disease, right? So the mutation will be lying there in this particular individual because it is coming in this family only, which it has other affected individuals. So this individual might still have the genotype for it, but it is basically not apparent phenotypically in this individual, that is individual 5 of generation 2, but it is apparent in in her children, right? So that is why now we need to calculate the penetrance. What is the formula for penetrance? I have mentioned in my other slide, but it will also give away the answer, so I'll just do it over here. So penetrance will be the actual number of people that are affected, which is how many? Oh, okay. Which is basically 1, 2, 3, 4, and 5. 5 people are actually affected, right? So we will write 5 over here. And the number of people who actually have this genotype that it should have been affected. So in case of an autosomal dominant disorder, we will speculate what the phenotype might have been. So over here, I write that according to me, this would have been AA, and this would have been a normal recessive individual, which is not carrying the disease, right? Then again, this individual might have been capital A, small a, because if it would have been capital A, this individual also might have been affected. Although it is a case of incomplete penetrance, so there is no sure way to know that, short way to know that. But this is what we are assuming, right? And this individual, again, the individuals that are marrying into the family are always considered as normal individuals unless and until it is stated otherwise in the pedigree problem, right? So these individuals who are marrying in are always going to be stated as normal individuals, right? Then this individual, we don't know whether or not it has incomplete penetrance or it is normal. So it is a question mark over here. Similar for this individual, right? But since they don't have any progeny further, we have no way of answering that. So we are just going to neglect these two individuals and we are going to assume that they are small a in their genotype, right? But this individual for sure, we know that it has at least a heterozygous, it is at least heterozygous for this particular autosomal dominant disorder. Why? Because its children have the disorder, right? So the children are showing this disorder with a normal mother. That is why they are heterozygous basically. Uh, okay. So over here again, this third one, we don't know whether or not it has the disorder and it is due to incomplete penetrance that it is not showing the disorder. But we are again going to consider these individuals as AA, right? Over here also, I'm going to slash off capital A, small a, and this is what we are going to consider these individuals. So now the number of individuals that actually have the disease, disease genotype are basically one, then two, three, four, five, and this other one, which is six, right? So six people have the genotype, but only five are affected. So this will give the percentage penetrance of this particular disease, and the answer will come out to be 83.3%, right? So the solution that I have mentioned, we can go through that also. It basically says that incomplete penetrance is a feature of autosomal dominant inheritance pattern. Okay. Then the formula for penetrance is basically penetrant divided by non-penetrant plus penetrant individuals, but percentage you can only find by this much, right? Then, um, okay, sorry, this should have been in another bracket over here. There should have been another bracket. Then we have actually solved this in the previous slide, and we know that the percent penetrance is basically 83.3% for this particular given problem. Okay. Then problem number 6 over here, which is another pedigree problem, says that Manu marries his cousin sister and gives birth to a child with cystic fibrosis. Now, cystic fibrosis is an autosomal recessive disorder, right? Given that Manu and his wife are normal, with one of their grandparents also showing the disease, what is the probability of his second child born to the couple to develop cystic fibrosis? So they have a they have common grandparents because Manu has basically married his cousin sister. So there has been consanguinity over here. I'm just introducing these terms or uh, basically speaking these terms so that you have a recollection of these terms also. So consanguinity is basically when you marry a relative or a blood relative or a close relative in your family as such. So over here it says, let me just switch to the pen. Here it says that Manu, who is a male, has basically married his cousin sister. So whenever consanguinity has to be shown, instead of a single married line, we use a double line, which was consanguinity over there, right? So he has basically married his cousin sister and given birth to a child with cystic fibrosis, right? We do not know the sex of this child, but this child basically has cystic fibrosis. This child is affected. But Manu and his wife are normal, and one of their grandparents also has the disease. What is the probability that the second child, the sex of the second child is also not known because this child has not been born as such as yet? So what is the probability that this child also have cystic fibrosis? That is what we have to find out, right? So we could list the entire pedigree, but it would also work if we are not just doing that. So it could be possible that, for example, their parents were somehow related, right? They came from the same family. and one of the grandparents were affected. So let us consider that the grandmother was the one who was affected by cystic fibrosis. So whose grandmother Manu and his cousin sister's grandmother were basically affected by cystic fibrosis, and their first child is also affected by cystic fibrosis, which basically means that both Manu and his wife are carriers, right? It might have been possible that their parents might have been carriers, and Manu's mother and his cousins or his wife's father might have been carriers, and they might have passed it on to their children, and Manu and his wife would also have been carriers, right? So now, since they are carriers, this particular child received the defective gene from both his parents, his or her parents, and that is why. Uh, just a moment. Okay. And that is why this child is affected. Now we have to find out whether or not this child will be affected, right? So what is the probability of this particular child being affected? I have not done the solution. So the answer for this is 1 by 4. How is it 1 by 4 is because there are two alleles that are present in an individual, right? So this particular individual, Manu, might have had this genotype, and his wife also might have this genotype, right? Because they were both heterozygous and carriers for the allele that causes cystic fibrosis. This particular child, if we draw a Punnett square for this, right? AA and AA, we all know what the probability is going to be, right? There are going to be two cases where the child might be a carrier, right? So 1 by 2 or half probability that the child might be a carrier, but that is not what has been asked. What has been asked is whether or not the second child will develop the disease. So what is the probability of the second child developing the disease? This is basically the probability, which is basically 1 by 4, right? There is one chance out of 4 that this child will have the disease. So this is the correct answer. Okay. Then we have a cloning question, and I saw in the discussion forum that people were pretty confused about what circular or linear cloning vectors were. I have included it in my lecture slide, but I'll explain a part of it over here also. So over here, what they basically say in the question is, we'll break down the question and then we'll read. Did not give me the laser pointer. Just a moment, guys. I'll have to. Okay, now I have. Huh. Okay, still not working. Anyway, we'll just move forward. Given below is a vector map of the plasmid PZAC, the size of which is 20 kb. So the entire size of this plasmid, as mentioned over here, is 20 kb, and the relative positions of the restriction site on the plasmid are indicated on the map, right? So they say that in the map given, you have the relative positions of restriction sites. For example, this BAM H1 site 1 and 2 has basically this 2 kb fragment between it. Then BAM H1 and EcoR1 has 4 kb fragment between it, and so on, right? So that is very visible. You have set up the following two restriction enzyme digestions for the plasmid. So these are two independent steps, digestions that they are asking us to do. The first independent question is, in tube A, digestion with restriction enzyme BAM H1, which is a single digestion, right? And in tube B, which is the second independent case is the digestions with restriction enzymes BAM H1 and EcoR1. Okay, with both these restriction enzymes, so that is why they are calling it a double digestion. Now we have to identify the correct option that provides the DNA fragments that you would visualize for tube A and tube B if you were to separate the digested DNA in an agarose gel. Okay, so now first what we have to do is restriction digestion, and then we will basically talk about the agarose gel DNA. And I'm unable to use this pointer, that is why I'll have to screen. I'm sharing my screen again. I hope it is still visible. Okay. So over here in the first tube, tube A, we are just digesting with BAM H1, which is a single digestion. So what is going to happen is that BAM H1, wherever it is present, it is going to basically produce restriction sites, that many restriction sites. It is going to cut at those many locations and it is going to produce those many restriction sites. So how many sites are going to be produced? One 2 kb fragment we'll get, one 4 plus 4 8 kb fragment we'll get, right? Then another 4 kb fragment we'll get and one 6 kb fragment we'll get. So we have to first see that for tube A, where are these options of 2, 4, 6, and 8 kb fragments, right? You understood how this will be 8 kb because these both fragments will be present between this BAM H1 digestion and this BAM H1 digestion, right? So, which option says that we'll get these two fragments? I think option number B is the only one that says that, right? Option A says 6 plus 4 plus 4 plus 2 kb fragments, which is incorrect. Option B says 8, 6, 4, and 2 kb fragment, which is what we found to be correct. Option C says 6, 2, and 12 kb, which is incorrect. And again, D says it will give 120 kb fragment, which is impossible because four restriction sites are present over here. It will not just 20 kb fragment would have been basically attained if there was one single restriction site, which would basically cut this particular circular DNA. With one cut, it would have become a linear DNA, and one linear 20 kb fragment would have been obtained. But this is not the case over here. Over here, these are the four fragments that we'll get. Now, I would like to use an eraser first of all, so that the second case would be discussed. Okay. In the second case, which is a double digestion, basically, what they are saying is that both BAM H1 and EcoR1 double digestion will occur, right? So it will cut on all these sides, 1, 2, 3, 4, and 5 sides it will cut, right? So what are the fragments that we will obtain? There is one 2 kb fragment, one 6 kb fragment, one 4 kb, another 4 kb, and another 4 kb fragment, right? So there is one 2 kb fragment, then one 6 kb fragment, and there are going to be three 4 kb fragments, right? Why am I writing it like this? Because when these fragments will be run in a agarose gel and basically in a gel electrophoresis reaction, how are these bands separated over there? On the basis of the molecular weight, right? So what will happen is that 2 B fragment will basically migrate the fastest, then 4 kb fragments will migrate, then 6 kb fragments will migrate in the gel, right? But all the 4 kb fragments will basically arrange on the same level because their size is the same. So they will be separated at the same level. They will give one band for all these three. It might be a thicker band than 2 kb or 6 kb, but it will be a single band, right? So over here, what we have to answer is that how many, sorry, what are the DNA fragments as visualized stabilized on an agarose gel, right? So on an agarose gel, 6 kb, 4 kb, and 2 kb fragments will be obtained in basically this double digestion. So B still remains the answer. But why is it so that in double digestion we don't have 3 4 kb bands or maybe 12 kb band because it is a 3 4 kb, don't have this earlier existing 8 kb band. I hope it is clear to everyone. Um, also hope that the meeting is still functioning, right? Because I have no idea whether I'm just blabbering or whether you people are able to hear it. I'm still going to continue with the meeting. If in case anybody faces a problem, please inform me. Okay. So the next question basically says that in a field survey of sunflower species, Ram identified a new variety of for the flower with violet petals and named it X. As the genotype for this trait was unknown, he has employed the following four methods in order to understand the dominant or recessive nature of the trait displayed by X. Based on your understanding, identify the option that lists the correct method. So, how will we identify whether or not what is the genotype of this the newly identified flower variety, right? So, which of these will work for him? Just a moment. Saptrishi, do you have a question?
Yes, ma'am. Good afternoon, ma'am. Sorry to interrupt you. Ma'am,
No problem.
actually, ma'am, I am unable to see the screen. So, ma'am, can you please share the screen once again?
Yeah, sure. That is what I was asking. If such a problem arises, you should intimate me as soon as possible so that everybody might be helped by that, right?
Now it's
Is
visible,
it
ma'am. Thank
visible
you.
now?
Thank you, ma'am. Thank you.
Okay. Uh, okay. So over here, from the options that are given, these four options that have been given over here, we have to identify which one might be the correct option or the correct way for Ram to identify the genotype of this new sunflower species that he has found, right? So the first option is a back cross between the species X to a genetically similar species. The second option is a back cross between species X to another species with an unknown genotype. The third option is a test cross between species X with a known homozygous recessive genotype. And the fourth option is a test cross between species X with a known homozygous dominant genotype. So the answer to this will be option C. Why is it so? Because only if they would have said test cross, a test cross basically means that you are crossing this particular individual from maybe the third, fourth, fifth, 10th generation to a parent which is homozygous recessive. And why are we saying the parent? Because we basically know the genotype of this individual and it even if it is not a parent, somebody of the similar species with a known homozygous recessive genotype is the only option for us to know whether or not this individual will have what kind of genotype will the individual have. Why? Because if we just for example, there was an individual X, right? We don't know its genotype and we test crossed it with a homozygous dominant maybe parent or individual, right? So when this person is crossed, for example, we'll take two cases. One, X had genotype capital A, and second case in which X had genotype capital A small a, right? Like these are the two examples I am going to take right now. Okay. So in the first case, if I cross this individual with this individual, we will have 100% individuals with what genotype? With a dominant genotype, right? Also, in the second case, if we like if you want a Punnett square for that to be drawn, I'll draw a rough one. So capital A, and capital A all will give capital A, right? So 100% dominant we'll obtain, and we will not know. Okay, just this much for now. In the second case, if we say that we are taking a test cross with a homozygous dominant for this case or this individual, what will we obtain? This is the Punnett square for what we'll obtain. The phenotype of these four will also still be dominant. So phenotypically, we will not be able to segregate whether or not it was homozygous dominant or homozygous or heterozygous dominant, right? But if we had taken, for example, a homozygous recessive parent, right, to perform these test crosses with, what would we have found? That in this first case, all the children would have been dominant, phenotypically dominant, right? On the contrary, in this second case, what would we have obtained? We would have basically obtained 50% children or 50% sunflower plants with one kind of phenotype and 50% with a recessive or the other kind of phenotype, right? So visually you can identify whether or not this particular individual or this particular unknown genotype species that Ram found would have been a homozygous dominant, heterozygous dominant, or if it would have been a recessive, then anyway you would have obtained 100% recessive kind of traits, right? But it is difficult to segregate between homozygous and heterozygous dominant, which is why we always run a test cross with a known homozygous recessive individual, right? So moving on to problem number 9. The below given pedigree shows an autosomal dominant inheritance pattern. Based on this information, identify the option that correctly mentions the genotypes for the individuals marked as 1, 2, and 3 respectively. So what kind of a pattern is it? It is a autosomal dominant pattern. Okay. It is an autosomal dominant pattern. All right. Now we have to identify what is this first, second, and third individual's genotype. So for third, we can safely say that this will be a small, since they are marking alleles as capital B and small b over here. So this third one has bound to be a small b, right? Why? Because it is unaffected, right? Since it is unaffected, it has bound to be small b. So it could be either option A or option B that could have been correct. Now let us see for individual 1 and 2 what are the probable options. So since this is an autosomal dominant inheritance pattern, right? And there is another thing that is linked with autosomal dominance, right? Which is what? Penetrance. But until and unless a problem or pedigree talks about penetrance, we are going to consider it as non-existent. We are just going to consider it as a normal problem, right? So over here, we say that this particular individual has the disease, but but we see that one of its sons are not affected, right? So what we could say is that if it would have been this, capital B condition, then at least one B from this parent would have transferred to all his children, right? So this individual would also have been affected, which means it was not a homozygous dominant, it was a heterozygous dominant individual, right? So this particular individual would have to be capital B, small b. So what are the options that list that? Option A and B are still in the running because both of them say that this particular individual has genotype capital B and small b. Then for individual 2, there could be another two possibilities. Either it could be capital B because it is affected, or it could be capital B, small b. And we have no basically method of finding out which one out of this it is, because the pedigree for this particular individual is not known. For this individual, this its parents and its siblings are basically mentioned, right? But for this particular individual, its parents or siblings information is not present. So we could say it could be either capital B or capital B, small b, because the individual is affected. So option A is correct because it says it could be capital B, small b, but option B says it will be small b, which is highly incorrect because then this individual would have been normal and it would not have been colored, right? So because it shows the disease, the correct genotypic information for individuals 1, 2, and 3 are that it will be capital B, small b. Okay, I just made a mistake in mentioning that it could be capital B over here. Sorry, I'm so sorry. This is not possible because once we see its progeny, right? Progeny, again, two people are unaffected. If it would have been capital B, then these individuals definitely would have been affected because even if one copy would have been present, it is an autosomal dominant disorder, it would have reflected over here, right? Since these two individuals are normal, it has to, without a doubt, be capital B, small b. So that was my fault. So the last problem from the previous year's assignments that we'll be dealing with is hair is a family affected with a rare skin disorder, right? A rare skin disorder which does not affect the survival or fertility. Review the pedigree and identify the mode of inheritance. So over here, could it be, let us first rule out certain inheritance, okay? Um, why we are basically going to rule them out is because it will make us easy. It is hard to basically write the genotype for all the possibilities, but it will be easier to write the genotypes for a few possibilities. So, what we can say is that females as well as males are affected in this pedigree, right? So, just a moment, pointer. Females as well as my pointer is not working. Let me see if हाँ ठीक है. Y-linked inheritance could not have been a possibility. We'll just rule this out. Why? Because if it would have been a Y-linked inheritance, only male individuals would have been affected, which is not the case. So it is not a Y-linked inheritance, right? Now let us see whether it could be an X-linked inheritance, right? So if it was to be an X-linked inheritance, we would basically, like, there is nothing that at once when you see this pedigree it can rule it out, right? It could still be an X-linked inheritance. For example, if you say that it is an X-linked inheritance, it would have to be a dominant inheritance maybe, right? uh, Let me just see. Since this female is affected, it would have at least one X with a defective allele, right? This XA we are considering as a defective allele, and this individual is normal, so this has a normal X and Y, right? Now, at least one X because it could not have been a dominant condition. If it were to be a dominant condition, then all the children would have been affected, right? So this individual has two children
Hello.
who are not affected. So it could not have been a X-linked dominant condition. It has to be an X-linked recessive condition.
Hello.
So it, yeah.
Yeah, ma'am. Your screen has been stopped sharing for last 5-10 minutes. I don't know exactly whether anyone can see or not, but I can't. Maybe I could be wrong.
Could somebody answer that? Uh, Just a moment. I'm also actually facing some problems since the past 10 minutes.
Yeah,
And
it's quite invisible actually.
okay. Was it not visible for anybody?
It was visible for me, ma'am.
Okay, but nonetheless, I'll share again. I was also facing some problems with the pointer. I don't know whether that was a this internet problem for me or for anybody of you, but I'll do it again, right? Maybe I'll pick up with this question
Yeah.
only for now, right? So this is the question that we were discussing, and up till now, what we had basically said was it could not have been Y-linked inheritance, right? This we can rule out. Why? Because both males and females are affected, right? Now, we wanted to check whether it could be X-linked inheritance. For X-linked inheritance, we were writing the genotype for these individuals, and we said basically that this female would have had at least one defective X gene, and not both of sorry, not both of her genes would have. Okay, I'm still facing problems. Okay, now I think it might work. Huh. And this male would have been X and Y, right? Because it is unaffected. And why would it not have been dominant? Because if this individual was basically dominant, then these children who are not affected over here would also have been affected, right? Because at least one X this person also receives, and at least one X this person also would have received from these defective X's that the mother had. So it is a, if it is an X-linked inheritance, it might have been a dominant, I'm so sorry. It might have been an X-linked dominant condition, but the female would have been heterozygous for this dominant allele, right? Then again, in this case, I feel that this would have been another heterozygous condition. This would have been a hemizygous condition. A male cannot be heterozygous for any X-linked disorder. This would have been a heterozygous condition again. This would again have been a hemizygous condition, right? And since the male basically passes it on to his female children and not his male children, other three are male, we could actually say that yes, this is an X-linked dominant inheritance, right? Because these female children are getting this only this X from the father, and this X is defective. So basically, these children, these female are also carrying the disease. And similarly, we can check over here also. This is basically a hemizygous male again, and a heterozygous female. Because it is a dominant disorder, it is still showing in the heterozygous condition. So all these will have the same genotype, right? If at all there is a confusion over here, you can ask. I'm just writing it monotonously because it is the exact same explanation for each of these genotypes, right? So from this, we can say that yes, it could be X-linked inheritance if it was X-linked dominant inheritance. So it has not been mentioned whether dominant or recessive. So X-linked inheritance could have been the option. But just because it says that it could have been X-linked inheritance, we can't really rule out the other options, right? We'll have to check it once at least. So for that, we are going to check for autosomal dominant because checking for penetrance is very hard, right? It will not be possible to check for penetrance unless and until there are some clear-cut examples. For example, and it will probably not be a this penetrant condition, because in case of a penetrant condition, there are certain characteristics that you see, which you also see in case of, say, recessive conditions, that the progeny is affected, but the parent is not affected, right? So in such a condition is not apparent over here. So what we'll check for is autosomal dominant. So just in the similar way that we wrote our pedigree for X-linked inheritance, could this be autosomal inheritance? For example, if we say that this is capital B and small b, right? It is an autosomal dominant condition, and this particular individual has small b, right? And because the female has capital B, small b, it has basically passed this capital B, small b to this individual, this capital B, small b to this capital B has been passed by the mother and the small b by the father, right? Similar is the case over here. These are all autosomes. So the male child could also be basically heterozygous for this condition, right? And the ones that are unaffected, they obviously have small b condition, right? And now if we see for the next generation, again, this capital B could have come from the mother in these affected individuals, and to the unaffected one, it might have given small b. And since the father only has small b to contribute, it might have only given the small b to his children, right? Similarly, this parent or the father might have contributed this capital B to these individuals who are affected, and to the others it might have contributed the other B, right? And the mother has only contributed the small b. This individual is marrying in, so we will always consider it to be normal and small b in this case. Again, since one of the parent is affected, the mother could have passed her capital B gene, which is defective over here, to her children. Small b they might have received from their father, and these children might have been normal. Similar for basically these children as well. They might have received capital B from their father, small b from their mother, and the others are all normal. So they will all be small b, right? So basically, when you write the genotype, it could have been autosomal dominant as well, right? And it is not without incomplete penetrance. Why? Because I gave you the reasoning for that. Because there are no such cases in which the children are affected, but their successors are not affected, right? So it does not include penetrance. It is not X-linked inheritance, but the answer to this particular question could either be autosomal dominant or X-linked inheritance, although in the previous year's solution, the answer to it was given as X-linked inheritance. But when I solved it, I basically found that autosomal could just as likely have been possible. So maybe you can also solve it once. Maybe I made some mistake. But this was the answer that was mentioned in the previous year's assignment. So that autosomal dominant I have added because like that is how I reasoned it out. It is very much possible. And if maybe somebody would just have seen this and ruled this out just on the basis of this being correct, it might have given them a loss in their assignment marks maybe, right? So you should check once. That brings us to the end of our first presentation for the day, which was discussing the previous year's assignment. I'll just close this, stop presenting this particular screen. I hope this particular part was clear to everybody. If at all you have any doubts, you can ask me right now. If you don't want to ask right now, you can maybe write to me. I'll try to get back to you. And for now, what we can do is we can move on to our second presentation for the day, which will basically be the lecture 2. Okay. So I'm starting to present my screen. I hope it is visible to everybody. So in this particular segment, what we are going to do is we are going to deal with concepts that were dealt with in week 2 and which I found out through the discussion forum that some of you were having a problem in understanding, and some concepts from my end that I feel should have been more elaborated, or you should have a working knowledge of those particular things. Okay, we are again moving or jumping right back into pedigree. This was basically the TA for the course had given this link, which is a very descriptive link if you want to know anything about constructing a pedigree. And it is a pretty professional kind of link. So I have basically kept two pedigree keys that I'm going to discuss, which were present on this link. Apart from that, if at all somebody is actually professionally creating a pedigree, there are certain instructions that need to be followed, which are basically again the keys that we are going to see. But these instructions, if we want it in a written format, they will just be that the key should contain all information which will be relevant to the interpretation of the pedigree. And for the clinical pedigrees, the name of the proband should be included. Family members' names should be and identification should be present. Then the name and title of person recording the pedigree. Historian is the person who is basically going to look after your or find out your family history. The date of intake and update, the reason for taking the pedigree, and the ancestry of both the maternal and paternal sides of the pedigree should be included if it is a clinically relevant pedigree, right? Then the recommended order of information would be basically this, which we don't really need to look in. And there will be a limit identifying information to maintain confidentiality and privacy. So these matters, obviously, like any other hospital policy, it is going to be confidential with between the patient and his historian or his consultant geneticist, right? So there are certain limits that have to be taken in that case also taken in mind. Just a moment. Okay. So if we see the pedigree key, right, we are just going to discuss the symbols. We are not going to discuss everything. Some of this might be very irrelevant for people. So the things that we are going to discuss is the symbols, right? The first one over here is male, which is represented as a square box. Female, which is represented as a circular a
Ma'am,
circle.
screen is not visible.
Okay, it's not visible. Okay, this one thing I'll keep in mind today also, that if at all I admit any, okay, my camera is also not visible. If at all I admit anybody to the meeting during my presentation, I will have to basically share my screen. Okay. I hope it is visible now. Okay. So, uh,
No, ma'am.
as we
It's
have
actually
talked
not
about,
visible, ma'am.
it's not visible.
No, ma'am.
Okay. Um, how about now?
Ma'am, from my side, I can't see it, ma'am.
Okay. Let me just do one thing. I will basically reopen that particular slide. Then I'll share my screen again. Okay, I'm sharing again. Hopefully it will be visible now. Um, Visible? Okay, I will continue in case it is not visible again, or it creates a problem. Please let me know. Okay, and I'll just take a break to have water. Okay, so as we have already discussed several times, a square will be used to present a male, a circle will be used to present a female, and a diamond shape will basically be used to present a person with an unspecified gender, or somebody whose gender we don't know, maybe an unborn child, right? Or somebody might not have been wanting to specify their identity or reveal their identity. That is why their gender is unknown over here. Then, if these individuals will basically be affected, then this circle or square or this diamond shape will be colored. If they have a partial like we have studied expressivity, right? That there could be varying degrees to which an individual might be affected. In that case, either it will be partially colored, or it might there might be different coloring patterns that are used in the same basically circle or square, which might signify that they have two or more conditions that are present, right? For example, somebody might have had hypertension and diabetes. These are very correlating conditions and they might run in the entire family, right? So we might we might color the circle or this square in such a way that basically it is apparent that okay, this individual had both diabetes and hypertension, this individual just had hypertension, and so on, right? If okay, we don't need to go through the third and fourth point. Then again, if there will be a slanting line that will be crossing this square or circle or a diamond shaped box, then it will mean that the individual has basically died, or it is a deceased individual. Not diseased, deceased. So there's a difference between the two. Deceased basically means dead, right? If this person might have had like a colored interior and a line through it, it might have mean that the person was diseased with the whatever factor we are talking about, or it was a diseased individual who has basically died, right? Then, if there is an arrow towards this particular square or circle, it means that this particular individual is the consultant, right? That means this is the particular person who has basically asked for this pedigree to be drawn, or who has basically consulted the geneticist or the historian over here, right? Then proband is basically again an individual, an index case only. It is going to again be signified by an arrow. But over here, the proband will be colored. It means that this is an individual because of which we have basically because this individual was diseased. It had this disease condition, or it had this genetic condition due to which we have basically started assessing the genetic history of the family, right? So this will be a diseased individual, which is an index case. Then, if it would have been a stillbirth, it will still show either the gender or the unspecified gender, diagonal slanting line across of it, and it will also basically list the week in which the stillbirth occurred. Along with this, if the karyotype is known, right, this week is basically the gestational age. Along with this, if at all the karyotype of this individual is known, it will be mentioned. For example, maybe it was a case of trisomy that did not basically complete its entire gestational period. So that trisomy 21 might be mentioned over here, right? So 47 to 1 or 47XXY, it might be mentioned over here. Then in case of pregnancy, you will basically mark the individual and the gestational age of the baby that that individual is carrying. If the pregnancy is basically not carried to term, there are certain, again, certain symbols for that that you will not see in the general pedigrees, which are basically spontaneous abortion, termination of pregnancy, and ectopic pregnancy. These are the symbols. You don't need to really know them. Apart from this, we need to know certain relationship lines, right? So I have a simpler version of relationship line also. So relationship line is going to basically mean what kind of relation an individual has with another individual, which is also basically shown in that pedigree, right? So over here, if we talk about the first relationship line between this man and this woman, there's a horizontal line that is connecting them. So this basically means that they are husband and wife, right? Then there is a line of descent which originates from between this husband and wife. So this means that when they mated, this is the line of descent, and this is a sibship line. So this means that people in this line will be siblings among themselves. And from the sibship line originate individual lines for these individual people, right? Now we can come back to this particular part, and this is again the same thing that is given in the first part. Apart from this, relationships could be basically denoted just like this through this relationship line or line of descent or subship line, right? Then consanguinity, as I mentioned in one of the problems that we solved earlier, is denoted by a double line between individuals, right? Then in the line of descent, which is that vertical or diagonal line, if the biological parents are shown, it will just be a vertical line of descent. If in case the individuals or the siblings are twins by any chance, right? They could either be monozygotic twins, that is, that after the fertilization has occurred from the same um, basically fertilized oocyte has broken down into two, and identical twins were born. So that would be signified by such a description in which there will be one line of descent, two individuals, and the two individual lines will also have another horizontal line that is joining it. But if it was a dizygotic twins, which is the fraternal twins, as we said, that there were two individual eggs which were basically fertilized by two individual sperms, and that is how these twins came to be. There will be no horizontal line between these individual lines of the individual people, right? If it is unknown whether the sibling, whether the twins are fraternal or maternal, there could basically be a question mark in between them. Similar for trizygotic individuals, we could basically just split it into three individual lines from the same line of descent, right? if the family history is unknown or unavailable, we basically just do not complete the individual line, and we just put a question mark on top of it. If there is no children by choice or some unknown reason, then basically that line will not be continued, right? The line of descent will basically have another horizontal line, which basically, for example, in reactions also, we either have reactions with an arrow end, or we have a reaction for such ongoing processes, or we have reactions like this, which means the reaction will not process, right? So proceed. That is seen over here. If it was by choice, sometimes it is mentioned that they don't have children because they have had vasectomy or tubectomy, right? If these individuals are infertile, it could be basically denoted by such a line. So there is a double line, right? Or if the actual specific reasons are known, then they could also be mentioned over here. If in case there has been an adoption and there is no biological parents that are present over here, then that has to be mentioned by a dotted line in the pedigree. Okay. So this was the pedigree part because somebody asked for the pedigree symbols to be explained. So that is why I thought maybe I'll explain some additional symbols also. Apart from this, this nomenclature, although sir has explained it in his lecture, but maybe it was not very well apparent to some people. So that is why I thought basically I kept the slide that I'll explain it once again. Okay, so for this first part, I first of all, I hope everybody is aware that these are two basically terms that I have picked from Sir's lecture itself, right? Where he was basically discussing some mutation, right? So this first one, which is C362G2A. What does this mean? G, this symbol and A. This basically means that there has been a transition in the coding sequence where a nucleotide has been changed from G to A, right? So C basically depicts the coding sequence, right? It says that there was a transition from G nucleotide to A nucleotide. And what does 362 determine? 362 basically means that this G that has transitioned to A now, right? This G that has transitioned to A was present at codon number 362. Right, and a nucleotide base number 362, which means that if this was the start site, AUG, right? It would have been 1, 2, 3, and so on. So at the 362nd position, right? This A was at position 1, U was at position 2, 3 was at position 3, G was at position 3. And at position 362, we originally had G, but now instead of G, we have A over there, right? This is what this particular statement means. The second one, which is P W121X, it basically means P stands for protein sequence, right? Protein sequence, which could also be called as polypeptide sequence or amino acid chain, right? So this basically means that in this protein sequence or amino acid sequence, at position 1 to 1, right? At position 1 to 1, that is 121 amino acid, which is basically a 3-codon thing, right? There has been a shift from W, which is basically tryptophan, right? It is the one letter code for tryptophan. W has basically, this was the amino acid that was present at the 121 position. But due to this particular change in the coding sequence, what has happened is that this particular amino acid has now been turned to a stop codon, which means X. X anywhere denotes stop codon. So in the protein coding sequence, amino acid number 21, which was earlier tryptophan, is now the stop codon. This is what is meant by W and X and 121 over here, right? So I hope that is clear. Uh, Moving further, okay, anticipation. So anticipation is a term that has already been discussed in classes. It is basically, what does it mean? It means that there are some disorders in which basically the subsequent generations, when if and when they get the disorder is more severe in its manifestation, and it manifests at a much earlier age than the original parent, right? For example, I have taken the most common example of anticipation, which is Huntington's disease or HD, as I have written over here. What happens in HD is basically there are trinucleotide repeats, right? What are trinucleotide? Trinucleotide basically stands for three nucleotide, right? These three nucleotides in case of Huntington are CAG. So this CAG is anyway, even in a normal individual, it is present in repeats, right? So there are going to be in a normal individual, which is represented by this green color over here, there are going to be less than 35 repeats. The number varies in literature, but approximately around like 30 to 35 repeats a normal individual has, right? So that it generally means that if this is a genomic DNA, we will have a segment which will have around 30 to 35 repeats of the CAG trinucleotide, which is generally required for making this Huntington protein in the individuals, and it is a normal condition, right? But sometimes what happens is the number of these repeats increases, right? Due to DNA, there is a phenomena known as slippage, right? I had thought of mentioning it in my slide. I had completely forgotten. Just now I remember that, okay, I will discuss this phenomena, but I will need some illustration to discuss it. Maybe in the upcoming lecture, I'll do it. So there is this phenomena known as DNA slippage, due to which sometimes the repeat number gets increased in individuals, right? So what happens is that if there are more number of repeats of this particular CAG, then it will manifest into disease condition. This disease is basically known as Huntington's disease. Okay, so this basically means that if the number of repeats increased to say 40, it will show an adult onset, right? But if there were around 120 or 121 repeats present, so since the number of repeats is more in that individual, obviously the onset of this disease will also be earlier. So it will convert into a juvenile's Huntington disorder. So this is what we basically anticipation is. So what could have happened is that there was an individual one. Huntington is mostly very common among females, right? So keeping that in mind, I'm drawing a pedigree in which there was this female and this male, right? So this female basically had Huntington disease, and she almost got the disease at the age of say 55, right? Then she has two daughters who are also affected, and they are also married, right? This first daughter got the disease at around 52, and this one got it at around 55 only. There was not much difference, right? When this particular individual has a child, right, she has a daughter and a son. Say that the daughter is affected, and the daughter got the disease at age of 40, right? And this daughter, by age of 40 also, you are generally married and you generally have children. So she had a daughter, right? And now this daughter is also affected by the disease, and this daughter is showing the disease at the age of 25. Say, for example, this daughter is showing the disease at the age of 25. So, what could have happened over here? That this person would probably have a normal parent, normal mother, right? Who would have normal number of CAG repeats, right? Then what would have happened? Maybe instead of 37 repeats in the mother, this particular daughter had around 45 repeats, right? That is why there was a late onset of the disease, right? Now, her daughters, the number of repeats might have increased because the repeats like they go up and down a little bit due to this process known as DNA slippage. So say that they anyway inherited around 45 uh, repeats from their mother, and in case of this daughter, the repeats went up to around 55, right? So she manifested the disease a little earlier than her mother, say 50 instead of 52, right? Now she has 55 number of repeats. She's going to give 55 number of repeats to her daughter also, right? And in the daughter, say that again, because this slippage is a continuous process, and the machinery, as we know, might already have been defective. That is why this slippage is occurring more and more. Now, over here, the repeats did not have to be increased from 35. It had to be increased only from 55, right? So 55 repeats were only already present. Now, say this daughter has 70 number of repeats, right? 70 is a very huge number. It is almost double the amount of repeats you should actually be having. So that is why the manifestation of disease was very early in this individual as compared to her grandmother, right? Who had the disease at the age of 55. Now, this individual's daughter already has 70 CAG repeats, right? And 270 CAG repeats, there might have been another bout of slippage, which might have converted into maybe 90 repeats in this particular individual, which is why she manifested the disease at the age of 25 only. Also, simultaneously, when we are talking about anticipation, that they are manifesting the disease at a relatively earlier age. How is that possible? I have just explained to you that basically the repeat number is changing, right? So the substrate for that repeat is anyway more for these particular individuals that are coming in the later generation of that Huntington disease family or hierarchy, right? So that is why they have disease that is manifesting more early in their life. Why is it manifesting early? Because obviously more number of repeats means more toxic protein that is being formed, which basically means that the severity of the disease will also be more. So this individual, by the time she turns maybe 30, she will have that much severity of the disease, which maybe her great grandmother had when she was maybe 70, right? So at the age of 30, she will manifest all those symptoms that her great grandmother might have manifested when this person was 70 years of age, right? Obviously, her lifespan will go further down. And like that is how anticipation works. So I hope it was clear to everybody. If not, please do ask, because I did find that a lot of people had doubt with anticipation in particular in the discussion forum. Moving ahead, this is another representation that I have given. In case of CAG length, CAG repeat length, above what basically the disease will manifest. So even if you have a slightly more number of CAG repeats, right? For example, 36 to 39 repeats if you were to have. you will still show the disease, but maybe the penetrance would also be reduced. Maybe you have the genotype for the disease or the genetic changes have already occurred, but maybe you are not showing that disease because the penetrance of this disease is reduced, or maybe even the expression level is reduced, right? Expressivity could also be different. And above 40 repeats, if it is present in the general population, it usually shows complete penetrance. Okay. Then moving forward with restriction enzymes. I'm so sorry. I just had a cough. Now I have a cold during this class. I just need to take a small break. Just a moment. Let me just clear my throat. I will share the presentation again. Okay. So next, I would like to discuss about restriction enzymes or restriction endonucleases. So these are basically one of the largest number of nucleases. So what is nuclease? Anything that is going to basically digest a nucleic acid. What are the nucleic acids that we know of? DNA and RNA. So one of the large number of nucleases that can cleave a DNA molecule at any site where a specific short sequence of nucleotide occurs. So these are extensively used in recombinant DNA technology. Also, different species of bacteria make different restriction nucleases, which can protect them from viruses by degrading incoming viral DNA. And each nuclease recognizes a specific sequence of 4 to 8 nucleotides in a DNA. So these are certain characteristics of restriction enzymes. So they say that restriction enzymes, did we come to actually find them? Because they are actually present in nature. They are present, basically made by a lot of bacterial cells. And what do bacteria do with these restriction enzymes? It is basically a defense mechanism for the bacteria, because the phage or the viruses that attack the bacteria, they basically introduce their genetic material, which is another some kind of nuclease, either RNA or DNA, into the bacterial cell. And what these restriction enzymes that are basically coded by these small green circles over here, what they do is that they go and bind on this incoming DNA or RNA, and they basically break or fragment this DNA or RNA, which basically protects the bacterial DNA, right? Also, there is another thing that I would like to mention over here, which is that okay, there's somebody waiting in the lobby. I will reshare my screen so that the problem that was happening earlier does not happen. Okay. So another thing that I wanted to mention over here was that these restriction enzymes, they will not be able to, how will they differentiate between nucleic acid of the host bacteria and the foreign nucleic acid, right? So they will only basically cleave the viral DNA or RNA. How will they know it is a viral DNA or RNA? Because it will lack these methylation tags, right? The bacterial DNA will have these methylation tags, which will basically signal it that, okay, I am this bacteria's own DNA. You can basically recognize me as a self DNA. You do not have to destroy me. This particular viral DNA will lack these methylation tags, which will basically aid its digestion by restriction enzymes. Okay, so moving forward, the types of restriction enzymes, these are certain examples that I have mentioned over here, which are HPA1, EcoR1, HIND3, and the PST1, right? So, as we can see over here, generally these restriction enzyme sequences or the sequences that are recognized by them, they are generally 6 base pairs long and palindromic, right? Earlier we read it could range from anywhere between 4 to 8 base pairs long. But another important property is that these sequences are palindromic. That means that it will read the same from both ends, from 5 dash to 3 dash in this direction and from 5 dash to 3 dash in the other direction. It is going to read exactly the same, right? Next, it says that restriction nucleases are often obtained from various species of bacteria, like we already talked about. So, the bacteria that are basically responsible for these particular restriction enzymes have been mentioned over here by me. Apart from this, another thing that I wanted to basically discuss was the nomenclature of restriction enzymes, because I feel we usually do it in the mol bio classes, molecular biology classes. So, but if we were even discussing restriction enzymes in a genetics class, I thought it might still be relevant to discuss the nomenclature of these restriction enzymes. So, for example, I take EcoR1, right? So EcoR1, the first letter as I have written it, is the genus name. So EcoR1 is derived from Escherichia coli, right? So E from the genus name is the first letter. Second and third letter come from the species name. So the first two letters of the species name, that is coli, basically give this eco, right? Then R, if this letter is present, the fourth letter, then it will be the strain name. For Eco R1, the strain was RY13, right? RY13. So that gives this R over here. And the Roman numeral ahead of it, which is 1 over here, it will basically be the order of isolation of this particular restriction enzyme from this particular bacteria, right? So, for example, if we take it for HIND3 or, for example, HPA1, which actually does not have a fourth letter or a strain name. So, HPA1, H comes from, H comes from Haemophilus, right? PA comes from Parainfluenzae, P and A, and 1 is its order of isolation. So it does not have a specific strain that it has been taken out from. It is just HPA1. Similarly, you can also try for HIND3 and PST1, right? It is one of the questions that is generally asked in competitive exams. So I thought of discussing it over here also. And that is just it for this slide. Apart from this, I also wanted to discuss what are the two kinds of restriction digestions that these restriction enzymes do, right? The first one is the blunt kind of digestion, and the other is creating staggered or cohesive ends. So what is a blunt digestion basically? For example, in case of HPA1, right, we are seeing that there is a sequence that it is recognizing, and this is the cut that this HPA1 is making, right? So what will be the resultant sequence? It will just be this, right? And CAA TTG, right? So this basically means that uh, these two sequences are present and it has a blunt end, right? As opposed to a staggered end. When I show you that end, maybe you'll understand what a blunt end means. For example, EcoR1 digestion. It will also basically recognize a similar sequence GAATTC, and it will basically cut over here after G, right? So what will it produce? It will produce one G over here and AATTC over here and one G over here and this CTTAA over here, right? So this basically means that it is present like this. So this kind of an end is known as a blunt end, and this kind of an end is known as a staggered or a sticky end, right? So why is it a sticky end? Because it is basically complementary to each other. This part is complementary to this part, so it has very high affinity to come back together very soon, right? And very easily. While this part, since it does not have any overhangs or staggered end, it does not, like, it will specifically require some or the other ligase function to come and combine back together, right? So these are the two types of ends, two types of restrictions that these restriction enzymes do. One, it creates either blunt or non-cohesive ends, or it could create staggered or sticky or cohesive ends, right? So both these kinds are used in molecular or recombinant DNA technology techniques. Um, Apart from this, we will discuss about very briefly about de novo DNA sequencing. We are also kind of running out of time. Hold on a moment. Okay. So, de novo DNA sequencing is somebody on the discussion forum again asked that how is it possible for us to basically bring up primers for like DNA sequencing if there is a PCR amplification step that is going to happen before the sequencing takes place. Some or the other primer might be required, right? But if the sequence that we are seeing is basically an unknown sequence, how is it possible that primers, how do we design primers? So basically what we do is we don't follow that particular step. Instead of that, what we do is de novo DNA sequencing, in which what is done is that the sequence of DNA is taken. It is basically mechanically or physically sheared down, and it is broken down into smaller, basically smaller segments, which are basically sent into cloning vectors, right? So as it has already been discussed in first classes, cloning vectors can efficiently take up only one such segment, and so one such segment in one such cloning vector can basically those vectors can just be given appropriate media, and it can multiply like anything. So amplification could be done through that process, and after that, sequencing is done, in which from which basically these reads are obtained. These reads, according to their overlapping areas, they could be arranged into contexts, right? These contexts are then checked for their directionality, whether or not it is forward facing, it should be reverse facing, forward facing, how it should be. According to that, it is basically generated into scaffolds, and later this scaffold, through a lot of uh, intelligence input and assistance. All this cannot be really done manually alone. So through a lot of input of these softwares, these scaffolds are basically then arranged into chromosomes. So it is a very tedious process, but this is how de novo sequencing is DNA sequencing is basically done. Then, since this came up, I also wanted to discuss that there were two other major sequencing techniques that I generally talked about. One is the traditional sequencing, which is Sanger sequencing, and the new one is the NGS or the next gen sequencing, right? So there are some differences between this, one of which is the sequencing volume. So Sanger sequencing is how basically the human genome project has been performed, right? And it has taken over a decade to basically obtain the final data from this technique. But whole genome sequencing is very efficiently possible in a very less time as compared to Sanger sequencing with NGS basically, right? Also, Sanger method only sequences a single DNA fragment at a time, whereas NGS is massively parallel. So how is it so quick? Because instead of sequencing a single DNA fragment at a time, it will parallelly sequence millions of fragments simultaneously in one run, right? And this process translates into sequencing hundreds to thousands of genes at one time. Also, NGS offers greater discovery power to detect novel or rare variants with deep sequencing. Also, I have put in an illustration for the two. So in Sanger method and NGS, one major difference is the extension step, right? In Sanger, only a single primer or a single template is used, but in NGS, multiple templates are used. And what happens is in Sanger, you get this template. First, the DNA is amplified, and then it is basically arranged. And it is another process that Sir has already talked about in his presentation. So you can see that how this arrangement happens, and then how detection occurs through readouts of the through the readouts of the fragments that we obtain. And were with respect to this, in NGS, what happens is, firstly, there are multiple templates that are involved. From these multiple templates, after the DNA is broken down, there are adapter molecules that ligate on small segments, particular segments, using which they are basically arranged on this array like plate. And after the DNA has been arranged on this array, basically the extension step occurs. So amplification occurs after this arrangement has taken place. And after that, basically, again, fluorophoric, DNTPs are added, and they are simultaneously detected and simultaneously logged for. So that is why NGS is very quick. You can maybe refer to some reading material and certain videos regarding what exactly NGS technique is. Apart from that, this was a very basic problem, but since somebody asked, what is homozygous versus heterozygous condition in case of genetics? So in homozygous conditions, basically, same alleles will be present on both the arms of the chromosome, right? Either it could be a dominant, this capital B, or small b, dominant allele or recessive allele, but it will be same on both halves of the chromosome, right? In case of heterozygous, both halves of the chromosome have different alleles. So it will be capital B, small b, or the reverse. But that is the condition where it is called heterozygous, when both alleles, both arms of the chromosome basically have different alleles. Okay, so I also wanted to discuss the difference between recessive condition and penetrance. Although I have put in pedigrees for both, I will very briefly discuss it because I think I have also given examples of it during the problem solving session. In case of recessive and penetrance, there are some common things that you will find. One of them is basically that there will be an affected progeny with unaffected parents, right? That is the case over here also in case of penetrance. So let me tell you, the left pedigree is basically recessive condition, and the right pedigree is the penetrance condition, right? So in the left pedigree, you can see that there are more such cases in which basically the individual is affected with unaffected parents, whereas there are lesser such appearances in case of penetrance, right? Also, I'm so sorry. Also, in case of penetrance, when you actually draw the pedigree, you will find out that like the genotype between recessive and penetrance actually varies. I thought of actually drawing the genotype over here, but if I do that, I will miss the subsequent slides. So maybe only if this question still persists, I will do it in the next class. Apart from that, I wanted to discuss the naturally occurring linear and circular DNA. So, why were we talking about these two kinds of vectors? Because DNA actually exists in these two forms in nature also. For example, this is the case in bacteriophage lambda, in which the same DNA has been shown in both the linear form as well as the circular form over here. In case of vector also, I have basically illustrated the example of the same exact vector in both its linear and circular form. If it would have been linear, it would have different kinds of restriction sites, which would be much different when these restriction digestions actually occur as compared to circular DNA vector. Which you might also have seen in Dr. Anshika's practice session, which was practice session 2 for week 2. I am also planning to actually make up problems similar to those and discuss it with you in the subsequent lectures. This was one of the template for that problem. Anyway, I will just be hurrying up with my presentation so that you can join the next class if you wish to. Uh, Another thing that I wanted to discuss was gain of function mutations and loss of function mutations. I will probably discuss it in my subsequent week's lecture also. I will keep the slide over there because I know people have reduced. The viewership has already reduced over here. And this is a concept that I want to explain to everybody, which is basically that I have basically taken example of cancer, which is a very talked about topic over here. And through that, I wanted to explain gain of function mutations and loss of function mutations. So as most of us might be aware, there are two kinds of cells that are basically talked about in cancer, right? One of these cells is oncogenes, right? So these oncogenes are the cancer cells. The other sorry before these cancerous cells are active, these are basically called as proto-oncogenes, right? These proto-oncogenes are cancer causing genes which could turn to oncogenes and actually cause cancer, right? So these proto-oncogenes are basically kind of silent. Due to certain mutations, they get converted to oncogenes. So they basically gain, this is a gain of function mutation. They are silent genes which gain a function and convert into oncogenes and cause cancer. But only this phenomena having occurred will not actually cause cancer. For cancer to actually occur, there has to be another set of genes that has to go wrong, which are basically the tumor suppressor genes. So, as the name suggests, tumor suppressor genes are basically going to be present to suppress the tumors, right? Even if this proto-oncogene converts into this oncogene, the tumor suppressor gene will function and it will basically suppress the cancer from occurring. But sometimes what happens is that this tumor suppressor gene, right, they will also basically undergo a mutation and they will lose their function, right? So loss of function will happen in this tumor suppressor gene, and this proto-oncogene has had a gain of function, and now the cancer gene is active and tumor suppressor gene is lost its function. So cancer will basically active, will basically occur, right? So tumor suppressor gene, as it is shown over here, act as brakes in the scar, right? And oncogene can basically act as some kind of a fuel. So we have a lot of fuel. This car is trying to race over here to cause cancer, but tumor suppressor genes are acting as those brakes and they are not letting this to happen, right? But if along with this gain of function, mutation, this loss of function mutation also occurs. Okay, if two such category of genes go wrong, then only cancer will manifest, right? So that was one of the examples that I wanted to share. Candidate gene selection, since it is anyway a topic for week 3, I'll probably discuss it in the subsequent class. And that is it for today's lecture. Okay, I will just get back to the meeting, and that is it, guys. Thank you for bearing with me throughout the lecture, and I'll meet you in the next week's lecture at the same time on Sunday, 3 to 5. So that is it. Thank you. You may leave. I'll have to save the recording. I'll have to stay back, but you may leave, right? Thank you for today's class.